It looks like it's time to start. So good morning, good afternoon, good evening, depending where you are in the world. Welcome to the second day and third session of the GARA 2020 webinar. Today's session consists of 4 talks and encompasses diagnostics, vaccines, and current status of next generation sequencing of African swine fever. For those of you that were not with us yesterday, this webinar was designed to keep momentum for GARA due to the postponement for us to have our annual meeting in Uganda that was supposed to happen in April 2020. However, as you know, due to COVID, this was postponed, and our next meeting will be August 24th to 26th in 2021 in Uganda. And hopefully everyone will be able to attend that meeting at that time. Some housekeeping rules is please keep your microphone muted at all times. If you have a question, please enter it into the chat box during or after the seminar. We're going to wait to ask all the questions to the speakers till the end of the seminar, and we'll ask them in the order they're received at the end. The seminar will be recorded and will be available on YouTube. And finally, I'd like to thank our Mrna and University of Pretoria team for hosting this webinar. Our first seminar will be by Dr. Sandra Blome, who is currently the head of the German National Reference Laboratory for ASF and CSF at the Frederick Loeffler Institute. Sandra has been at FLI for the past 12 years, where her research has focused on studies on pathogenesis of viral infections, diseases with particular emphasis on virus-host interaction and diagnostics vaccine development. Sandra will now give her seminar an update on diagnostics for African swine fever.
So, hello everybody, I hope you can hear me. So I will give you an update on diagnostic challenges, but hearing yesterday's webinars, I decided to restructure my talk a bit because I heard a lot of very basic questions, and I decided to give you an overview and also to discuss then the challenges we perceive. Why do we need laboratory diagnosis for African swine fever? There is definitely a problem in a clinical diagnosis. So differentiation of African and classical swine fever is mainly impossible based on clinical signs alone. And the same is probably true for several other differential diagnoses. I just brought you this picture. On the left-hand side, you see domestic fattening pigs at the 7th days post-infection with the highly virulent ASFV strain Armenia. So this is the one that is, causing our headache. And on the right-hand side, you see domestic pigs and also some wild boar at the 7th day post-inoculation with a highly virulent CSF strain. The only thing that you see is that the animals are sick. They are having fever, they shiver, they don't eat, but in the end, it could be any other disease. So in the end, laboratory diagnosis and confirmation is a mandatory thing. What possibilities do we have to detect the virus? First, what we call direct methods will detect the pathogen itself. So you see, African swine fever is a really beautiful virus, but it is very complex. And we can detect either the infectious virus, some viral antigens, or also the viral genome. So it's, in the end, the sequencing or the sequence. Then, of course, the host will react to the virus, and we can also detect the responses of our hosts, so antibody responses, and in some cases also T-cell responses. So one of the crucial things, and also one of our challenges, is whom to sample. We definitely need somebody to raise suspicion and to take the right samples from the right animal at the right time. And of course, somebody should also send the sample in an appropriate way. Sometimes I receive samples that are in various stages of decay. This is not a very nice sample. Some tests will still work, but there are others they won't. Let's have a look at the first picture. So on the left corner, in the upper corner, there is a group of pigs, 5 pigs. All have been inoculated, but only 4 are infected. If you see this animal sitting there in the middle, this pig will probably come to you eating your boots. It's easy to get a sample, but this animal is definitely not the right one to sample. At that time point, it was completely negative. And if you would have taken only this animal, you would have done the wrong diagnosis. So remember, ASF is not necessarily a highly contagious disease. So it does not spread like hell. So you have to sample the risky ones.
Spread like what? Hell.
So the risky ones are the ones with fever. They show runting, just the sick ones. Let's have a look at the right-hand side in the lower corner. So in the pigs that are in the back of the picture. The one that is on the right side, this pig will probably not stand up. It's lying in the corner of the pen. Yes, it's the one that you have to go, the sample, and you have to go there. It may be deep in the shed, but this is the right one. It has high fever. It looks like a moribund animal, and it also has a high viral load. In this case, you detect the disease. So keep in mind, the best test cannot perform with inappropriate samples. And if you take the wrong animal, this can be the case. In the lower corner, I put a picture that gives you some headache. These animals have high fever. It's 18 days post-infection, but they look rather healthy. They all went to you, and you have to make a choice. In this case, you just have to do random sampling. These animals have an acute transient infection, and these animals have virus and antibodies. So if you try to sample them, you will find out that they at least saw the virus. What samples could we take? If you sample sick or fallen animals, you are in the end targeting direct methods. In this case, we would like to have EDTA blood samples. Please avoid heparin samples if PCR is intended. Heparin is a strong inhibitor of PCR reactions, and don't send this if you like to do a PCR. There's also a possibility to use serum. I will come back to this later. We have to be very careful because in the very early and late phases of infection, serum is much less suitable than EDTA blood. The genome load is low in these cases. Another good sample is a spleen. Spleen is very high, or has high viral loads, and if you can get it, it's one of the most suitable samples. There are also other tissues that you can send. It's lymph nodes, the tonsil, lung, kidney. So it's a systemic disease. So in the end, I will find the virus in most tissues. There is actually, in the end, recommendation to use bone marrow for white boar if they are in a better shape. The problem is that bone marrow is not a good sample if you sample the fatty bone marrow. You really need the red one to have good diagnosis. So whenever you can find something else, please avoid to send us, in the end, bones with the bone marrow. Then, of course, you can also do the indirect methods. They are mainly used for screening purposes, for monitoring, looking in affected holdings to give you an idea about the age of the infection. In this case, serum, plasma, and to a certain extent, also meat juice and whole blood can be used for some applications. One important message is, if the first choice sample is not possible, just talk to the lab. There is a lot of options in most of the laboratories, but please ask them what they can handle. What are the methods we are going to use? If we want to detect the infectious virus, there's a possibility to isolate the virus mainly on primary cells. So in the end, on macrophages derived from bone marrow, from lung, or from blood. And we usually do hemoadsorption assays. But as some of the isolates will not do this, they don't have an hemoadsorption capacity, we can also stain macrophages and look with immunofluorescence. There are still some countries that do bioassays, but in terms of animal welfare, we should probably skip bioassays as much as possible. If we want to detect the viral antigen, there is a possibility to stain cryosections. There is an antigen ELISA, but please keep in mind that this is not very sensitive, and it also has problems if the animal has antibodies. And then we have now also the possibility to use lateral flow assays, but again, I will come back to them. They have a low sensitivity. To detect viral genome, we can use different approaches of PCR, gel-based PCR, real-time PCR. I put this in red and bold because this is the main method if we talk about field diagnostics or diagnostic from field. Then sequencing, some other applications like direct PCR and isothermal methods. And antibody detection, the first line of defense is an ELISA, but we also do for confirmatory reasons, indirect immunoperoxidase test, indirect fluorescence antibody tests, immunoblots, and also lateral flow assays. We are not just dealing with something that where we don't have rules. There are a lot of rules, and on a global scale, you can just see the OIE. Just see this link below. There you will find the terrestrial manual, and you will find all information there. So in the end, if you look at this broad range of possibilities, we do not really have a challenge in diagnostic tests. ASF has a lot of problems, but diagnostics is not the worst. Look at the real-time PCR. Real-time PCR has a high sensitivity and specificity, and it can be used with a large variety of sample matrices. Reliable detection is possible from roughly 3 to 4 days post-infection, and it's also possible very long, so roughly 70 to 90 days. If you look at some drawbacks, it's rather expensive and requires good equipment. We have now commercial tests available, and most are suitable. This is an advantage. These tests are really validated, and often they are licensed. So it's quite good to use them also in labs that have not been accredited or they are in the process of accreditation. For these tests, nucleic acid extraction is needed, and this can be a very crucial point. The nucleic acid extraction can be done manually or automated. So in the end, PCR is suitable for large-scale testing, and also extraction can be done large-scale. You should prefer tests with an internal control. It can be either heterologous or endogenous, but these internal controls secure reliability of negative results. The quality control is towards inhibitors of PCR and or the sample quality. But high sensitivity also means that you have a high risk of contamination. You should always check plausibility of results and have a backup system available in the lab. Backup systems should target another genomic region and are only used for confirmatory testing. If you're dealing or you're working in the lab, keep your fingers off these confirmatory tests. You only take them off the fridge when you have a problem. Just to give you an example, I heard yesterday how to interpret these CQ values. If you have a sample with a high viral load, you have a low threshold cycle because the fluorescence is rising soon. So if you have then a low viral load, it's the other way around, you have a high threshold cycle. If you have a PCR with a control, for a positive sample, you have a fluorescent signal for the target, but the internal control can be negative. This is because we restrict the PCR resources. So the primer concentrations are much lower for the internal control. The target is served first. So if there's a lot of target, the internal control can be negative. It does not have to, but it can. Negative samples should have no signal for the target, but a positive signal for the internal control. An invalid sample with a lot of inhibitors will have no signal. I just put it as a panel on the right side. You see the red one is the positive one with the blue curve for the target, a green curve for the control. So both worked in this case. Then you have the green box over there. There you only have the internal control, no target, it's a negative sample. And you have one with just baseline calling. This is an invalid sample. It could be inhibitors, it could be that you made a mistake, but this one is invalid. The real-time PCR is in principle quantifiable, so quantification is possible using standards. I just put in the middle an example of the standard curve. So you could find out how many genome copies are in your sample. The question is, for animal disease control, this is not really important. Even a few copies will render the animal positive. So it is mainly for research purposes and for the purpose of trials. The antibody ELISA is the other first line of defense. Detection of an antigen-antibody reaction is the case with labeled antibodies. The labeling is done with an enzyme. It's often horseradish peroxidase, and we measure optical density at a defined wavelength later to define if a sample is positive or negative. Despite the fact that we get some semi-quantitative results, it's a rather qualitative outcome. These commercial tests that are available are usually fit for purpose, but they show a moderate sensitivity and specificity, and for that reason, confirmatory testing is beneficial. Most tests work with serum and plasma. Some have also been validated for filter papers, swab punches, and also meat juice. In the end, you have to check what they are meant for, and they can give reliable results. The detection is usually from roughly 10 days post-infection, and it lasts for at least several months. It's rather low tech, but I had already some problems, not only in Africa, but also in Balkan states, that the water quality was low, and then we had a lot of problems with the buffers, and the optical instruments can be crucial. If they have not been validated for 30 years, there is definitely a problem with the reliability of the results. There are indirect and competitive formats on the market, and these systems use different antigens. This also means do not expect in all cases to have the same result. Check plausibility and also check what antigens they are using. An important thing is, what can we expect to detect? Whenever you choose a sample or test priority, keep the infection biology and pathogenesis in mind. There was a lot of discussion when the first antibody positive wild boar were detected in Poland. But what does it mean? It only means that the animals survive longer than 10 days. There is no really predictive value to it. You also should keep in mind that ASF is originally an arbovirus. So if you want to detect the virus, blood contains the highest viral loads, and 1 drop of blood is much more infectious than loads of secretions. Viremia is rather long, even in the convalescent animals, and a lot of discussions about carriers and so on is rather a semantic issue. What we can say is that most of our animals, at least all animals in my hands, recovered or died, but the phase of detection can be rather long. You saw 90 days. If you want to do the first line of defense, so early warning, choose a sick pig to obtain initial samples. And keep in mind, viral genome is not infectious virus. So if you find a few genome copies on water or wherever, this does not mean it's highly infectious. I brought you some figures from recent animal trials. Just look at the right-hand side. There is the qPCR results of EDTA blood. And on the top, you will find the percentage of positivity. You see that between day 7 and 40 all animals were positive for viral genome, and then the percent positivity decreased to roughly 50% until day 90. Day 90 is rather long, but we also followed the animals further on, and there was no detection at 164 days post-infection. So these animals can still recover completely. And on the right-hand side, you see the comparison of EDTA or the qPCR from EDTA blood and virus isolation, in this case from sera. And you see that the virus was gone by day 60, at least 63, but the viral genome was still detectable. So whenever you look at the test results, keep this in mind that this is not the same. I brought you some slides also from a very recent animal trial to show you that already at day 4, we have a reliable detection from blood, but also from quite a lot of tissues. So in the end, roughly day 3, day 4, this is when we can detect the viral genome. Look at alternative samples. Shedding with secretions and excretions is rather low, and we have to discuss if the suitability of matrices is always the case. Swabs may not help with small holdings or individual animals because this can really give the false reactions, but they may be fit for purpose in large holdings. If you look for a herd-based result or when sampling dead animals, it can be that it is fit for purpose. There is actually some studies from Belgium where they show that nasal swabs from fallen wild boar give very reliable results. This contrasts a bit what I show here, but in the end, if an animal is dead and you have a lot of blood everywhere, this is not really a problem to detect than the viral genome from swabs. And it's always the case, hemorrhagic signs will aid detection, so bloody fecal or oropharyngeal swabs will also be possible. In the end, when you talk about what samples to take, you should first do the optimal. But if the optimal is not possible under your circumstances, there's a lot of options that can still give you some reliable results. Look at the antibodies. Antibodies appear roughly between days 7 and 14 post-infection. But as I already mentioned, they do not have predictive value for final disease outcome. So it is not a problem to find a dead wild boar that has both antibodies and viral genome. It only tells you that this animal survived longer than roughly 10 days. And for this reason, the antibodies will tell you about the age of infection and can give you very valuable hints about the high-risk period. And they can also detect infections with strains of lower virulence that probably even went unnoticed. Most of the cases that we have at the moment with genotype 2 strains are highly virulent cases. But still, you have seen these animals that were sick but were not showing too many signs. This kind of things can go unnoticed, and in these cases, you will find antibodies. I just wanted to go back to sample matrices a bit. This was a recent trial that we did, and we compared EDTA blood and serum in qPCR. In the end, serum is a very nice sample matrix because you can use it in any of your tests. You can use it for the serology, you can use it for virus detection. It's a clean sample if you are lucky for the virus isolation. But have a look at the graph. On the left-hand side, it's 4 days post-infection, and we compare the viral loads in genome copies for EDTA blood and serum. And there we see a huge difference. Just look at this blue animal on the left-hand side. For EDTA blood, it's 400 genome copies. It's already low, but look at the serum, it only has 3 genome copies. And if you then consider pooling, you may miss this early phase of infection. So keep in mind, it's a disease that can be transmitted by ticks. The virus is attaching to erythrocytes. And in the initial phase and later phase, the serum contains much less viral genome than EDTA blood or whole blood with the erythrocytes. You see on day 7, all the animals were sick. There's no problem in detection also in serum samples. So when you use samples from sick animals, it's probably not a problem. If you are going to test animals, for example, in contact holdings, be aware of this problem and aware of these discrepancies. We often heard, we couldn't send you a sample because it was of bad quality. This is a bad excuse, actually. In the very beginning, we tried to do storage and sample quality testing or assessment. What we did is we used different matrices. In this case, it's German, so I will read it to you. It's blood, spleen, muscle, and feces. And we kept it for 1, 2, 3 months at different temperatures that you have available in the lab. It's the fridge, the freezer, and the incubator. And in the end, I just put there these red squares. For the blood at day 0, it was even lower in genome load than the sample that we took after 3 months at 37 degrees centigrade. So please don't tell me that the sample is too bad. It's usually a bad excuse. In Europe, we target the wild boar carcasses, and sampling is definitely not easy. If you ask the hunters to sample them, the rotten carcasses are smelly and to a certain degree disgusting. Some organs are not available anymore. If you look at these half taken or half eaten carcasses, the spleen is no longer there. Blood or decent fluid is not available, and some carcasses are just skeletons. Against this background, we tested a lot of alternative sampling methods. Our colleagues from the Netherlands have tested bone marrow. In the end, how to get the bone marrow out of the bones. We have tested blood swabs, and I will share a bit of the results just to show you that it is sometimes a bit of creativity to find a way to still get decent samples. What we did is we used swabs that we could handle in the lab, and we used these swabs for both antibody and genome detection. To cut a long story short, you can say, okay, you lose a little bit of sensitivity, but we have a good correlation between our values of these genotube swabs and the EDTA blood. The same is true for the antibody detection. You can also use these swabs for other diseases if you do syndromic surveillance. And you always have to discuss fitness for purpose. Is it really a problem if you detect one wild boar less in an affected region? Because there's not only one wild boar showing the disease. So in the end, this is something you have to discuss with your risk managers, your veterinary services, but you should keep in mind that fitness for purpose is not always 100% sensitivity and specificity. You can combine to a certain extent these methods even with some point of care diagnostics. We just did it in the beginning for fun because we had the possibility, but it turned out to be much better than expected. We actually cut pieces from the swabs and used it in an ELISA that was licensed for the use with filter papers. And you see that you even have some doubtful cases that were doubtful in the ELISA that also give doubtful results on the lateral flow assay. In this case, it's a lateral flow assay for the detection of antibodies. Please keep it in mind. The antibody LFD is rather good. What about this point of care testing? Can we use it in the field? Antibody LFDs are quite sensitive and highly specific. But the problem is the target animals, the fallen animals, may not have antibodies because we are in early warning looking at passive surveillance. So they are rather nice, but maybe not the right choice. The antigen LFDs lack mostly sensitivity, but they are also specific. In our hands, sick pigs are usually detected under experimental conditions, but more data are needed to assess their suitability under field conditions. Dick Pfeiffer said yesterday in his presentation that we have to be open-minded and do not disregard this kind of options. In the end, we have to have more data to really say if they are fit for purpose, but definitely they will not detect all animals. There are also some field deployable genome detection methods that are under development and evaluation, and future will show their true value. There is one problem with these point of care tests. Who is going to test? And who is going to report to whom? And what is the impact of positive and also negative results? In my opinion, a positive result is, in the end, there's enough evidence that there is problem in the farm. But a negative result could still mean there's disease. So in the end, these animals have to be retested, at least if there was a suspicion based on clinical signs. For Germany, this was, of course, my target. I have problems to see the application. If you give it to the farmer, what will he do? And how long will this test be in the shed before he uses it, and so on? So the quality assurance is a problem, and also the reporting of the results. But anyway, we have to be open-minded and also consider this kind of test systems. I was often asked, should and could we test meat and other matrices? Other matrices including crops, including whatever surfaces. We try to find a way, and in the end, there is no major problem with products that have retained their structure, and also salami with even additives, cheese, pepper, whatever. The question is rather, is it something we should do? Because in the end, we do not know the impact of these findings, and how would you follow it up? If you find somebody at the airport that is bringing a sausage and the sausage is positive, what does it tell you and what will you do? In the end, it's a rather expensive assay, and we have to also see what is the follow-up. In the end, if you really want to test it, there can be applications. We have found out that magnetic bead-based extraction methods are suitable for most matrices. There are inhibitors present at a lower level, but they do not exclude the diagnosis. So keep in mind what you will do with the results. But in the end, yeah, it's possible. Some words about quality and impact. As I said, the fully validated commercial tests can facilitate diagnosis. The labs should still do some verification with in-house samples at the national and regional lab. Actually, there is rather a problem with result interpretation than test performance. Whiteboard samples can be tested with all available test systems. However, the sample quality matters, but this is not a problem with whiteboard. It would be also a problem with domestic pigs. And please keep in mind that some discrepancies result just from biology. So please check the background, check plausibility, and do not just take figures for granted. The use of real-time PCRs should implement also an internal control to obtain reliable data, especially with poor samples. Do not forget the confirmatory testing. There are several labs that also do Stone Age testing, virus isolation, and so on. Please don't forget them. A PCR is a good thing, but you will never get the virus. Keep fitness for purpose in mind. In the end, it means that you do not have to detect a single genome copy under all circumstances. There are a lot of examples that you can control a disease with suboptimal test systems. If you look at bovine herpes virus, for example, some of the DIVA tests has a sensitivity mainly of 80% and also not really a good specificity, but it was helpful to control the disease. Keep also the impact and significance in mind. You cannot, for example, test a large batch of wheat for freedom from ASFV. If you have a positive result, it tells you something, but if you have a negative, you may have just taken the wrong sample. And there are some points of discussion. They are related to the pooling of samples. I am a strong defender of pooling. If you pool 5 samples, I think it can be done. But however, you have to establish workflows in your labs. And also there's a lot of questions about the point of care tests. So in the end, there's a bottom line. Tests will only generate results. You will get a lot of figures, but the challenge lies with sample choice and interpretation, especially interpretation. And this in the end needs experts and also experience. So please ask the peers, also ask some from other countries that had already ASF in previous decades. And please always do plausibility checks. The last slide, I want to share the beauty of diagnostic assays with you. This actually is an ham absorption assay. So it shows you that the virus grew in the samples. These are infected macrophages that collected more or less erythrocytes. They look a bit like raspberries. So you see, diagnostic assays can be very beautiful. And with this, I thank for your attention.
Thank you, Sandra. Does anyone have any questions? They can post them in the chat.
I stunned them, no questions.
Okay, we got one question. How much blood do you recommend to take for field samples?
It depends actually on how much you can get. For the PCR, I need 100 microliters. But in the end, if you can send 1 tube, we can do most of the assays. 1 tube meaning it's 7 to 9 milliliters.
What advice do you have for bone sampling?
The main problem with the bone sampling is really you should only apply it when there's a skeleton. If we have a bone, please take the femur. The femur was best suited to obtain bone marrow that we can still use in diagnostic assays. So this was the one. It's not so easy to open it, but it contained most virus. So in the end, this is what I would recommend, to use to send the whole bone to the lab. So in the end, you extract the bone, you put it in a suitable device and send it as it is to the lab, and the lab is going to open it. They don't love this, actually.
Sandra, are the qPCR commercial kits based on European strain sequence, or do they also include African strains?
This depends a bit on the manufacturer, but the ones that are licensed in my lab, they all also included African strains. They are detecting most strains that are available, at least through GenBank. There may be some exotic strains that will not be detected, but in the end, they are they have been tested also with these genotypes.
The commercial test for antibodies have moderate low sensitivity and specificity. If a positive on the competitive ELISA, what confirmation test do you recommend?
In my hands, we first use indirect immunoperoxidase assays, but we also use immunoblot. But the one that is the first line of confirmation is the immunoperoxidase test. It is true for most of the European countries.
Okay, how do you combine the diagnostics for ASF and CSF and other diseases?
In the end, what we do is we do extraction of nucleic acids. So we will have DNA and RNA, and we can then use it for both PCRs. And usually the samples are also suitable to do the serology for both. Actually, this was the reason why we, in the first go, went for the serum, because the confirmatory testing for CSF in our hands is a neutralization assay. That's a cell culture-based technique, and plasma is killing your cells. So this was the reason why we went for serum in the first place. It can be used, as I showed you, but it has some drawbacks.
What was the largest amount of time post-mortem, estimated number of days between death and sample collection, you witnessed in a wild boar sample that tested positive?
It's difficult to tell because to tell the age of a carcass is not so easy. Some of our colleagues, Carolina Probst and others, have tried to develop guidelines how old the carcass is that you see in the field, but the decay is very much dependent on ambient temperature, on humidity, and so on. So it is not so easy. But we know this for sure. There has been a study in Lithuania where they excavated in the end wild boar carcasses, and they were still positive for viral genome after 400 days. We talk about viral genome, we're not talking about virus.
Do Spsc values vary between strains?
For some assays, it will probably do. For most of the PCR assays, I did not see too much of a difference. Some assays have some problems with genotype 9 and 10 viruses, but they still detect them. But there's a bit of a difference. For the antibody ELISA, I can only use what I have. I'm rather restricted in what I have done under experimental conditions. There I did not see problems, but I'm not sure about the whole African situation.
What is the best way to preserve tissue sample for virus isolation in case of no availability to a -80 freezer?
Without a freezer. I mean, if you use the spleen sample cooled, I have a rather nice chance to get the virus. So if you can keep it cool, it is at least a good opportunity. We did actually some field-based trials with one of my colleagues that accompanied the excavation. We tried to put it into PBS just to have it in a rather sterile liquid. In this case, we did not find the virus, but the sample looked well.
It says alternative to virus isolation using fresh PPIS.
Sorry, I did not get the question.
It says alternative to virus isolation using fresh, that's PPIS. I'm not sure what that is. I don't know, Mrna, if you want to clarify.
Can you explain? Because
we'll skip that and see if we can get what that means. Which virus proteins do you suggest to use in antibody tests?
In the end, what we see is in the commercial assays, there is in the end P72, the P30 and P62 as antigens. You know, the nomenclature is not so easy, so it can be P73 or it can be P72, 32. But in the end, these are the main antigens. 54 is also one of the antigens. What I use in my hands is still a lot based on the P72, and as an earlier antigen, the P30.
Okay, alternative to using virus isolation, using fresh PPLS.
I don't know. I haven't compared, so I'm not completely sure. I'm sorry.
Do you think at some point you are going to be doing sequencing on the field using nanopore technology, Mrna and Oxford Nanopore?
In the end, we have done some studies with Mrna, and probably Jan will talk about it. If you really deploy it in the field, you should ask him. In the end, the best results were done on PCR products or on very or the long DNA. So if you don't have it too much fragmented, I could imagine that it is field deployable in the future, but you should ask Jan later.
We got our last question is, can you expand a little more into pen-sign devices as an option for genome detection in the field? Also, any experience with nanopore sequencing?
My own experience in this direction is rather limited. So we have done our own studies with NALF detection. So using LAMP assays combined with nucleic acid lateral flow assays. It worked in principle. The main problem was that there was a problem with contamination. When you did it the one day, whatever you tested the next day was positive. So this was a serious problem. Probably you could overcome it, but it would mean reestablishing workflows and so on. In my hands, LAMP assays and other isothermal methods did not perform as good as the PCR. But I know that people that really went into details may have different opinions. But however, for me, it was not an option. There is now a lot of machines that are field deployable for PCR, including some from TetraCore, from Indical. There are different on the market now, and they give good results, even the ones that do direct PCR. But again, direct PCR is very contamination prone, and we have to be careful when interpreting the results. But I see some hope and future for this kind of assays.
Okay, thank you, Sandra. We're going to move on to our next speaker. It's an excellent talk. Thank you.
Thank you. So, bye.
Okay, our next seminar will be by Dr. Manuel Borka, who for almost 20 years has been the lead scientist for African swine fever and classical swine fever at Plum Island Animal Disease Center. And prior to his current position, he was part of the original African swine fever program at Plum Island in the early to mid-90s. His research has focused on identification of determinants of virulence, pathogenesis, and vaccine development for both African swine fever and classical swine fever viruses. Manuel will now give his seminar on an update on live attenuated vaccines. I think you're muted, Manuel.
Now you can hear me?
Yep, now I can hear you.
Okay, sorry. If there is anything that everybody that working in vaccine development in African swine fever agree is that animals that are inoculated with live attenuated strain of viruses are protected against the challenge, at least with the homologous isolate. The point here is how we produce those live attenuated vaccines. And let me just, for the purpose of classifying, let me present three different ways to produce this virus. One is by just using field isolate that has low virulence. Second is by attenuating the virus by culture passages. So the virus, when they get adapted to the virus, pass it to the culture, lose the virulence. And the third method is by genetically engineer viruses that by deleting specific genes, they became attenuated. I'm going to go through all these three classifications, and I will give some examples, and I give some also some examples of the advantage and disadvantage of each of them. Let's start with the low virulent field isolate. This having several examples of people working with this. I just pick one. It's a paper from Mibus and Ardiri from Plum Island back in 1980. And this person, what they did is they used Brazilian and Dominican Republic isolate that were not completely virulent, and they inoculate animals. And what happened is that the surviving animals, they were challenged with highly virulent virus from the same region, and they get protected. They even went a little bit further, and they used Lipon-16 as a kind of heterologous challenge, and still they got some degree of protection. So, and studies like that have been for a while, and actually from this experiment, there were 2 or 3 different viruses that were all natural isolate that start being used as a model to study the production of protections and the mechanisms of induction of immunity in African swine fever. And some of these viruses, I will talk a little bit of some of them, are like the isolate or TADA3. I think this isolate was isolated from soft ticks in Portugal. This is a non-absorbing virus. And interesting, this virus produces a strong protection when it's used to immunize animals, and the animals are challenged with an homologous virus that is called ORT88.1. And the problem with this virus is that, like in many others that are natural isolates, that they still have some residual virulence. And there have been work done, and I will refer one from the group of Linda Dixon, where they tried to get rid of this residual virulence by deleting specific genes that have been previously described as involved in the virulence. And I'm talking specifically in the case of this paper of 2 genes that were removed, that were what have been demonstrated before, that attenuate Spanish isolate. I'm talking about the removal of NL and UK genes. But what Linda did is that she produced a virus that is called delta DP2. And what happened is that this virus is still, after the deletion of these genes, still present a residual virulence. And also, and the worst scenario was, and that is happening, that is very frequent, and it happened to us in Plamaila also, once that the genes were deleted, they not only didn't show elimination of the residual virulence, but also there were a clear decrease in the ability of the T88 virus to induce protection. So genetic manipulation, even in those natural isolates, attenuate isolates, had to be a little bit careful. Another example of these low virulent isolates are the NHP68, that is also a non-absorbing virus isolate in Portugal. And that interesting, half of the animals that were inoculated with this virus present some kind of chronic form of disease. And the surviving pigs, or the pigs that were not heavily affected, actually were very, very well protected with the challenge with an homologous isolate, the Lisbon 60. And because that, as in the case of the ORT88, this model also became a very good system to study immunity and mechanisms of protections against the disease. As I said, the virus also has a strong level of residual virulence, and in a paper from the laboratory of Yolanda Rorisha, they tried the same thing that they tried Linda with the ORT88, that is, get rid of that level of residual virulence. And what she did, actually, and the group, is to delete 4 different genes that were previously shown to be involved in viral-host interaction and in the immunomodulation of the host. And unfortunately, what they see is that in all the cases, they still see presence of residual virulence in several of the pigs that were inoculated, either with the wild-type virus, with the parental virus, or with the recombinant virus. An interesting issue is that all of them, in this case, they were protected when they were challenged with Lisbon 60. And an interesting result in this paper was that animals that were inoculated with the parental virus, the NHP68 attenuated, and they were challenged with Lisbon 60. Interesting, when they were challenged with Armenia of 07, that is a derivative of the Xerxes virus, they were also protected. So that was an interesting presence of crop protection that I thought that was interesting to point it out. Now, again, the problem with this virus, as the other one, was the residual virulence that cannot be easily solved, the problem that cannot be easily solved. The last virus that I'm going to give as an example is also a natural isolate, also non-absorbing. That is a virus that have been isolated and worked by the group of Dr. Jose Manuel Sanchez Vizcaíno in Madrid. And this is basically a virus that was isolated in Latvia from a wild boar. And what this group show is that oral vaccination with this virus protects over 90% of the wild boars that were challenged with the virulent Armenia 07. And this results the authors proposed that this could be a vaccine that can be used in while bored. But there is also a paper that has been published by the same group where they inoculate this virus to domestic pigs, and what happened is that both animals in some way develop residual virulence, develop same signs of disease that were a mild disease, but they were disease. And also when they put 4 animals in contact with these 2 directly inoculated animals, half of them, 2, also developed sign of the disease. So the presence of residual virulence was there. The paper was finished by showing that 1 pig that was directly inoculated and 1 pig that was infected by contact, they successfully survived the challenge with the Armenia virus. My point on this is that we have to be a little bit careful with the low virulence field isolates, since the problem of remaining residual virulence. And I think that should be a point of concern in the use of this virus as a potential vaccines in the field. The second group of way to produce attenuated strain of virus was by passing the virus in tissue cultures. And that's something that there are several examples out there. I just picked one that is a paper from Ruiz Gonzalo back in 1986, where the person worked for a long time in Spain. What he did is pick several field isolates in Spain that were temporally and geographically related, and they passed those isolates in Vero cells. And what he saw is that by passing the virus in Vero cells, they became attenuated. When he put those attenuated virus in pigs resist the infection. And not only that, the pigs resist the challenge when the challenge was done with the parental non-adapted virus. He also went a little bit further and trying to see if there were some kind of heterologous protection among these different viruses. And although they were, as I said before, temporary and geographically related, there were not really levels of cross protection. But there was a good example of what the attenuation of virus can be done by passing through culture. I recommend that a paper that had been published by Dennis Kolbasov and his group, that actually is a review that contains all the efforts of his institute of producing producing virus adaptation, and actually we should say attenuation of virus by adaptations in PBMC and other cell lines. And actually, is for those that are interested in this particular theme, I think it's an excellent reading to see how much work these people have done in order to develop what particularly I'm trying to make an example here: the attenuation of virus by passages in cell culture. We have done some of that experience in Plumine, and we try to adapt the Shorsia virus to Vero cells. And the original idea was, among other things, trying to study what is the correlation of the genetic changes with the process of adaptation of the virus to grow in Vero cells, but also trying to see if we were able to produce a vaccine by this methodology. Unfortunately, we failed. Once that we got our virus attenuated, the attenuated virus was not able to produce protection against the challenge. But still, we learned something that I think that was important. That is the huge genomic changes that happen in the virus while the virus is being adapted by passing the virus in Vero cells. And this data that is, I would say, an extreme example of gene modification during cell adaptation, we have seen that with other viruses and with other cells. So I would say that the problem of using virus adaptation are the dramatic genomic changes that are involved in this adaptation. And I would say that I will be very careful in handling this virus and attenuating virus in this way, because in our experience, the incorporation of deletions and genetic modifications, it doesn't stop. We have done this with virus, Shorsia virus adapted for over 130 passages in Vero cells, and we still, in passage 130, we still can see the incorporation of new mutations and new pieces of deletion. So I think it's something that need to be considered if some of the viruses wanted to be used as a vaccine in the field. So the last group is the viruses that are genetically engineered. And I will bring here a cartoon, very old cartoon, I think that was put together by Laszlo Zak and John Nealon at the time that they were working in Plum Island under the direction of Dr. Dan Rock. And basically what these guys did was something that was very important. That is, they developed a methodology to produce genetic modification, gene deletions in field isolate, highly virulent field isolate of African swine fever. Why this was important? Well, because it was the only way to really study the effect of the contribution of a particular gene to the process of producing the disease in a natural host. By doing so, by first time, it was possible to identify genes that are important in the process of virulence, in the process of producing the disease. 3 genes were at that time are identified as such: NL in Spanish isolate, UK also in the same Spanish isolate. I think there was C70, and the 9GL gene in the Malawi isolate. The deletion of 9GL was also done in another isolates like a Pretoria, and they also produce attenuation of the virus. The interesting part of this is that all these attenuated strains, they were able, when inoculated in pigs, to solidly protect against the presence of the disease when the challenges were performed using the parental virus. So these 3 papers were the basis to produce recombinant virus by genetic manipulations and produce viruses that can be used as a vaccine candidate and protect against the parental virus. And I will not talk about the genome of the virus. Everybody that knows that it's very big, 150 plus genes. The only thing that I would like to mention now is that very few genes out of the 150 plus have been actually really studied experimentally. Most of the functions are ascribed by prediction, but not by real studies in the bench. And I would say that probably one of the best ways to study the function of the gene is by deleting the gene and see how the deletion of the genes alter the phenotype of the virus, in vitro or in vivo. And last year, Linda Dixon presented a paper that I think that was very interesting because a very good review. And basically she put a table where she identified all the viruses that have been produced by deleting a specific genes or group of genes by different labs. And what I did is I added to the data in this particular table, the data that came in the last year. And basically, we can summarize that 19 different genes have been tested by this methodology. And out of these 19, 8 of those had been genes that were associated in one way or another to the process of virulence in the animal. What I'm trying to say is that these deletions of these genes or group of genes produce attenuation of the corresponding strains, and those strains could be used as a potential vaccine candidate. And I would say that everybody that work with recombinant African swine fever know that this is an extremely, extremely time-consuming, money-consuming, effort-consuming process. But when I see these numbers and I say, okay, out of 19 viruses that were produced, almost half of them show that there are viruses, genes that are related with attenuation and can be used for producing a vaccine. Actually, I think that this very tough work is very, very rewarding. And what I'm going to do is to pass a quick review of the papers that have been published in the last 5 years that are related in the production of recombinant viruses by recombinant virus that can be used as a vaccine candidate. The first virus that was produced in the context of Georgia virus was the one that we did by deleting in 9GL. And we published this in 2015. Basically, the virus was attenuated up to 10 to the 3. When we put higher doses than that, the animals die. And that was a very important lesson that we learned, that is not always the same deletion of the same gene, even if the gene is very highly conserved, produce the same effect when done in a different strain. As you see here, the same amount of virus when deleting 9GL in Malawi, animals survive perfectly fine. Actually, have been published before that you can put 10 to the 6 of the 9GL Malawi and nothing happens. So we learn a lesson here. The interesting is that the animals that were inoculated with sublethal doses of 9GL were solely protected against the presence of the disease, not of the presence of replication of challenge virus, because some of the animals still produce the virus, the challenge virus replicate, but there were solid protection against the presence of the disease. Lessons learned, 9GL is had to be used in this particular case at sublethal doses. And the other lesson is that, as I said before, not the same deletion and different genotype background produce exactly the same result. And we have seen that later with other examples. The next group of genes that were focused was a group of mutation family 360-505 that map in the left region of the genome of the virus. And we were interested in this region for several reasons. First, this region is deleted in several natural attenuates like the NHP60A or HORT-ADA3. So they could have some relationship with attenuation. Deletion of these areas were associated with the changes in cell tropism, as have been shown, by the group Dan Rock in tick cells. Importantly, several groups have shown that these genes have a good effect in immunomodulating swine immune response, especially the innate. And also important, a paper from John Nealon a long time ago showed that he can rescue with this particular area of the genome the virulence of an attenuated virus by recombination. So all of those things put our attention in this particular area, and we produce a virus that we published in 2016, where we delete 6 genes of this particular area, 3 of the 360 family, 3 of the 505 family. And actually the virus, when they put in pigs, even at 10 to the 4, they were completely attenuated. And the challenge with the virus in the way that we do challenge, that's it, 10 to the 3 intramuscular animals were completely protected against the presence of the disease, but not always against the presence of viral replication. And this very interesting, Linda Dixon came with a paper a few months after ours, where she published the deletion of a larger area in this particular region of the genome, comprising 9 genes this time, the same 9 genes that are affected in the natural isolate HORT-ADA3. And she showed that the virus that she produced using Benin as a parental virus was completely attenuated when it was put in pigs, and all the pigs that received this Benin Delta mutation family were also completely protected against the challenge. There were some replication of the virus after the challenge, but there was a solid demonstration that the deletion of this area produce an attenuation and can be used as a potential vaccine production. Interesting, there was a paper just coming in this year from our colleagues in China, Dr. Wu's group, where actually this paper, what this scientists did is to took all the information that was out there about genes that were involved in production of attenuation, and they make all those deletions in using as a template the virus that they isolate in China. So I'm going to refer to this paper several times in the rest of the talk. But basically right now, what I wanted to show is that this group produced also a virus that was extremely similar to the one that we produced in Plum Island. And this virus that they call 6GD, because there are 6 genes deleted, were completely attenuated, as in the case of our virus at Plum Island. And they were animals inoculated with this virus were solidly protected against the challenge with the parental virulent virus. Interesting, and this is the first time that anybody does this with recombinant virus in African swine fever. They make a reversion to virulent experiment. They pass this virus 5 times successively in group of pigs. And what they saw is that in the 5th passage, 1 pig presents signs of the disease. And when they took the virus from 1 pig, and that pig in the 5th passage, they put in a new group. Now all the pigs show temperature, and 1 pig showed a very important presence of the disease. I couldn't find in the paper the genomic changes that explain the reversion of virulence. So the genomic changes on the virus isolated in that feed passage. And I think that that will be extremely important to understand how come that the virus that had illicit genes now became virulent again. It will be extremely informative in terms of understanding virulence in African swine fever, know which were the genetic changes that support that reversion to virulence. And this is just the summary of the mutants produced by deleting this particular area. Although it's not chronologically ordered, that is what I try to do here, I would like to talk about another virus that was published in this particular paper by Dr. Bush's group, that is the 7GD. 7GD virus is basically the multi-gene family deleted virus plus the deletion of the CD2. In the same paper, they showed that deletion of CD2 does not alter strongly the virulence of the virus. Some of the animals survive, and this lack of effectivity of deleting CD2 as a method for producing attenuation concurs with the data that we produce in Georgia virus by deleting CD2, where we show that by different routes and by different doses, the deletion of CD2 doesn't alter at all the virulence of the Georgia virus. So going back to the 7GD virus, they show, of course, that were completely attenuated when they put in pigs. And interestingly, still, the animals get protected when they were challenged with the virulent virus. More interesting is that the 7GD virus remained attenuated after the 5 passages, where the 6GD failed in pigs. And also was important, it was innocuous when they were inoculated in pregnant souls. And I believe that this virus is now being used in larger study, including much more bigger numbers of pigs. So I think that it will be very interesting to know what is the status of those results once that they are published. One thing that brought my attention lately is a paper from the PBRE group where they show something that was not tested before. That is how long the immunity can last in animals that have been inoculated with an attenuated strain. They use 2 particular viruses, ORF88 and Benin multi-gene family. And what they show is that animals inoculated with these 2 viruses, 130 days post immunization, they did not have protection. That was very, very interesting. According or with the same result, the paper that I just mentioned, Dr. Bush's paper, they show that 7GD also after 80 days after vaccination, the animals are not 100% protected. They are only 80%, and the titers of antibody are lower. So it seems that there is a problem in the length of the immunity produced by this attenuated virus. This, I think, is a very interesting topic. My only concern is, since these 3 viruses have been produced, the attenuation of these 3 viruses are produced by deletion of one particular area, that is the multi-gene family. My question is if that happened with viruses that are attenuated by using deletion of other areas. And that is something that we are trying to prove right now at Plum Island, using some of our viruses that are not related with the deletion of multi-gene. I wanted to move ahead, and I wanted to talk about the virus that was also published, I think, in 2017 by the group of Dr. Fernando Rodriguez in Barcelona. And basically what Fernando published is a virus that is a BA71 isolate that has deleted the CD2. That is not the only genetic modification that this virus has, if not, because there is some also small changes to make some of the virus inducible. So there are some other alteration of the genome of the virus, but basically the main alteration is the deletion of CD2. They showed that this virus animals inoculated with this virus was completely protected. And interesting, when they were challenged with the homologous virus, they found that the animals were protected against the challenge. More interesting is that they showed that the animals also were protected when they were challenged with a slightly heterologous virus, E75. Remember that the parental virus is Badajoz. And more interesting is that the group of pigs that were inoculated with the BA71 delta CD2 also resist the challenge with Georgia virus. So this paper brought two important points here. One is that the presence of heterologous protection, something that is not very common out there. And the second, that is very important, this virus grows in a stable cell line. I think this virus grows very well in host cells, what it will enormously facilitate the transition of this virus to the industry. I would like to just mention another virus that we produce when we have problem with producing the stability of the 9CL virus in Georgia. We produce a new virus that is the 9CL with the addition and deletion of one gene, UK, that we knew that were attenuated. The addition of the UK attenuates some strain of virus. We showed that deletion of UK in Georgia by itself does not attenuate the virus, but when we put together the deletion of 9CL and UK, we obtain a virus that was completely attenuated even when put in doses of 10 to the 6. All the animals inoculated with 10 to the 4 or higher were protected against the challenge. And interesting, by first time, we showed that protection can be achieved only 14 days after the inoculation of the vaccine in African swine fever. So this virus that we are pretty happy with that, because the stability in terms of attenuation and the ability to induce protection, even early protection, and we're still working with them and trying to push ahead as a vaccine candidate. Another interesting piece of information is that in Dr. Booth's paper, they also produce a virus that has a 9CL deletion and a UK deletion. Of course, the virus that they produced was attenuated in animals, but the problem was that this virus, the 9CL UK, the version from Dr. Booth's lab, did not protect against the challenge. And that was something that really surprised us, because we tested several times at Plum Island, our version of 9CL UK, and we couldn't understand very well what could be the differences. And still, we don't understand. We're working a little bit on that. But a brief analysis of the data that published by Dr. Booth show that the deletion of the 9CL gene in the way that they did in Harbin is wider than our deletion. And the reason why our deletion is concentrated in the middle of the genome was to try to avoid the alteration of the genes that are flanking the 9CL, especially B962L gene that is predicted to be an RNA helicase gene. And what we try to do in our deletion is to respect the promoter area of that gene. That's something that is not based in the data published by Dr. Booth. So we believe that probably this deletion alter the transcription of the B962L gene. We don't know if this is exactly the reason why they obtain different levels of protection than us in Plum Island. This is something that we're working in our lab to see if we can show that that is probably the difference. Another thing that was, or another piece of information that was very interesting was published by Linda Dixon by deleting one particular gene, DP148 gene. This gene never had been previously worked in the past, and she used to delete that in the Benin system. And what she showed is the genes map in the right area of the genome of African swine fever. And what she showed is that when the gene is deleted in the Benin strain, they are complete attenuation of the virus. Not only that, if not that, also challenged with Benin virus, in those animals inoculated with the mutant, they were completely protected. Interesting, the protection is achieved even if the animals are inoculated with the virus by intramuscular route or by orally administration, what is very important in terms of thinking administration of vaccine in the field. Dr. Wu also produced a version of this virus, and actually what he found is that the Chinese strain of virus, the deletion of TP148 does not alter yes, the virulence of the virus. So the virus became virulent, and so we are not very sure why, if the difference in the result between Linda group and Dr. Booth's group, but I think that is another nice piece of information showing differential effect of deletions in different backgrounds. And finally, to close this talk, I would like to talk about the last virus that we produce in our lab, that is the deletion of a gene called I177L, again, a gene that had not been previously described. And this gene also mapped in the right area of the genome. And what we did is we reproduced a virus that on the Yorkshire strain. And the first thing that we saw is that the ability of the virus to grow in macrophages is severely affected after the deletion. Still, when we put in pigs, even a high dose of 10 to the 6, the animals were completely healthy. So the virus lost completely the ability to produce virulence. And all these animals were solidly protected against the challenge with the Yorkshire parental virus. We have big expectation in this virus because, again, induced protection even working at very low doses, 10 to the 2, and lacks residual virulence even working at high doses like 10 to the 6. Another thing that they didn't mention is that it does not induce virus shedding. Animals that are cohabitating with animals that are inoculated with even 10 to the 6 for as long as 1 month does not get the virus from the vaccinated animals. And also something that is very important, at least for us, that is induced sterile immunity. And I say this because this is the first candidate that we have with developing Blome that protects not only against the presence of signs of the disease, presence of the clinical form of the disease, but also when given in the right dose, protect against the replication of the challenge virus. I would like to finish this talk with some gaps analysis. Things that I think that should be done. There are several candidates out there, and I think that we should study critical aspects of this candidate, like onset of duration and duration of immunity, genetic stability, attenuation of that stability. How stable that attenuation is, calculation of minimal protected dose, and also study the possible route of inoculation. Though not all the candidates that are out there, actually, I would say that most of the candidates out there, including us, we don't have answer for all these questions. I think it's very important to standardize the methodology for testing vaccine efficacy. It's amazing when you go through all the papers and the amount of variables that are there is amazing, starting from the type of animal that we use, the dose of the vaccine, the route of the vaccine, and very, very important, the challenge, how we challenge our performance in terms of how much virus it's using the challenge. There are a huge heterogeneity in that, and I think that we should try to standardize a little bit that. I think it's time to start developing vaccine candidates that has the capability. I think it's important also to start investigating the basis why there are differential effect or deletion of one particular gene, even if the gene is very well conserved, when the deletion is made in different isolate. And I think that this is not only academically important, if not that it's important in order to create knowledge to when we had to do this kind of deletion on new strain of virus, we will know which deletion, which genes are more stable in terms of producing the same phenotype. Finally, I think that we should continue discovery and characterization of virus associated with virulence. I think that this is the basis of showing where we are right now. I think that the huge amount of investment that we means all the lab working in virulence in African swine fever had done are the basis of why we are now close to have something that can be considered as a vaccine, and that is probably why commercial partners are start being interested in helping and pushing these vaccine candidates out there and make of those real vaccines to be used in the field. Sorry if I went over a little bit in time. I appreciate the opportunity to give this talk. Thank you.
Thank you, Manuel. We have one question already. It says, live attenuate vaccines seem to protect pigs if homologous. Are these vaccinated and surviving pigs still infectious for other non-vaccinated pigs?
I think that that's well, this is part of the question that we don't know. We don't have the answer. I think that one thing that I learned working for almost more than 20 years with this virus is that you better do the experiment and then you answer the questions. I really don't know. The possibility is there, of course.
What are the prospects for live attenuated vaccines to induce heterologous immunity? Is it just the right genes have not been deleted, or will a cocktail be needed? Maybe GARA should play a role in recommending standards across labs.
I think in the last slide, I'm trying to answer that. We don't know what are the basis of cross protection. We don't know what are antigens that are involved in the process of cross protection. So I think at this point, everything had to do by trial and error. Probably in the next talk, when they were going to talk about Subunit vaccine, we can put some more light on this. But basically, right now, I think everything had to be answered by trial and error. You got your virus, and you make a test, cross protection test, and see what happens. And this is not only in African swine fever. You go to the fundamental world, it's exactly the same. It's very difficult to predict cross protection from just a sequence. You need to have the actual data.
What is the status of a good cell line for growing Georgia for production?
I don't know. There are papers there that say that there are a nice paper published by Linda Dixon, where they show that by using ZMAX cells, they grow Georgia virus. I do not know any other cell line that have been published right now that support the growth of Georgia virus.
Does the Delta I177L inoculated pigs have joint swelling?
We never observe, in the type of experiment that we conduct, we never see any negative clinical aspect of the inoculation of 177L. Can
Delta I177L mutant be produced stably in cell culture?
We are working cell culture. I imagine cell lines, right? The cell culture are primary macrophage, yes, of course. On the cell line, that is an issue that we are working right now in our lab.
Can ASF live attenuate vaccines recombine or regain their virulence?
Well, that is why I think it's so important, the data from our colleagues in China about the 6GD virus that revert after the fifth passage. I think that the data, maybe I was unable to find the data in the paper, but I think that the genetic information regarding what happened in that virus, it will be extremely important to understand reversion to virulence. To my knowledge, this is the first reversion to virulence that have been shown in any recombinant virus. But it's true also that it's the first trial where a reversion to virulence experiment has been really conducted. So
MA104 cell line is possible for vaccine production?
No. That cell line worked very well for the detection of infectious particles from using different isolates, but does not support efficiently the replication of the virus in large scale.
It doesn't look like there's any more questions. So.
Okay.
Thank you, Manuel. Very nice talk.
Thank you. Yeah, thank you.
We have about 2 minutes before the next talk, so we'll start in 2 minutes. It looks like it's time. Our next seminar will be from Recombinant, Portugal, who finished her PhD in 2008 on modulation of apoptosis by African swine fever, and has worked in Portugal and Germany on different areas of ASF research, namely methods for ASF recombinant virus selection, genome sequencing, interferon signaling, and viral gene expression. In 2017, she joined the Pirbright Institute as a postdoctoral scientist in the ASF group, where she has been involved in the development of effective vaccine for African swine fever. Rekul will now give a seminar on an update on subunit vaccines.
Oh, hello, everyone. Can you hear me?
Yep, we can hear you.
You can hear me. Okay, good. So I will be talking, if I can share my screen. Yes.
不，这就是那篇文章了，这篇那个，就是这篇那个。
Oh, okay. Someone didn't mute the microphone. So I will be talking about an update on the current status of subunit vaccines for African swine fever virus. First, what are these subunit vaccines according to the World Health Organization? This type of vaccines do not have live components of the pathogen, only a fragment or antigenic parts that elicit a protective immune response. Antigenic properties of the various potential subunits of a pathogen must be examined in detail to determine which particular combinations will produce an effective immune response within the correct pathway. And often a response can be elicited, but this is no guarantee that the immunological memory will be formed in the correct manner. However, these type of vaccines are, of course, safer and easier to produce and generally more stable than live attenuated vaccines. However, they may not induce strong protective immune responses, and in many situations, adjuvants are needed. As we all know, African swine fever is currently still spreading in the world, and which is a big problem because we don't have a vaccine. And as Manuel has mentioned before and talked extensively about this, protection can be conferred by attenuated strains. However, these pose safety problems, and protection is limited mostly to related virulent strains. There are some cases of cross-protection against heterologous strains, but this is rare and still not well defined. Subunit vaccines, on the other hand, show promise, but have had limited success so far for ASF protection. But there is some protection that is induced using some strategies of immunization, which leads us to think that optimization and using the right combination of antigens and delivery systems might work in the future. On the other hand, also, the use of these subunit vaccines allows us maybe to direct the immune response toward the correct pathway that we know may provide protection. So I will be talking about different experiments, different immunizations that have been made along the years for ASF using protein antigens, DNA, and viral vectors. It will not be an extensive description, of course, but I will try to show the work that has been done that shows most promising or interesting results. So the first antigen, the first subunit vaccines that have been used were based on protein antigens from ASFV. During the 70s and 80s, a lot of the proteins from ASFV have been identified, and it was evident that antibody responses were present in serum from immune animals against these proteins. And sometimes also there could be neutralizing activity from this serum, preventing infection either in vitro in cell culture, but also in vivo by passive transfer to naive animals, or even piglets being fed with colostrum from immune sows. One of the first experiment using a protein as a subunit vaccine was made by Ruiz Gonçalves and Cole in '93, where the authors saw that there was a hemaggglutinating activity in the supernatant of ASF infected cells. And the authors saw that this hemagglutinin that was released from the infected cells could be separated from virus, and it was a glycosylated protein, which was later on identified as the CD2 homologue of ASF, involved in hemadsorption. The authors purified this hemagglutinin from the supernatant and used it in immunization of just 2 animals, but they could see that one of the animals was protected against challenge with virulent virus. Later on, the same authors cloned the sequence of this hemagglutinin for expression in baculovirus, and used this baculovirus expression system to immunize 3 pigs. They could see that there was induction of specific antibodies inhibiting hemagglutination, and also in one of the pigs that had received the highest dose of the vaccine or of the immunogen was capable. The serum of this pig was also capable of inhibiting the infection in vitro. When the animals were challenged, all 3 animals survived infection with virulent virus, and the pig that had been immunized with the highest dose even showed no viremia. The other 2 pigs showed viremia, but this viremia was cleared later on in the experiment. So these HA-induced antibodies could have a role in protection, or they could induce other types of protection. So this set the quest for other viral proteins that could also induce protection, and especially viral proteins that could be target of neutralization. So produce antibodies that could neutralize virus infection. In '93, Zach tested different antibodies and for capacity to neutralize infection in vitro, and he could see that all the antibodies that could neutralize infection also immunoprecipitated a protein of 72 kilodaltons, which was P72, the capsid protein. Gomez Puertas et al. In '96 also used these 3 of the highest immunogenic proteins of ASF, P72, P54, and P30, and he expressed these proteins in E. coli or baculovirus. The recombinant proteins were then used to immunize pigs, and the serum of these pigs was tested for ASF neutralization assays, and it was seen that they could neutralize the infection in vitro in cell culture quite extensively, above 70%. The recombinant proteins were also used to purify antibodies from serum of convalescent pigs by affinity purification. And these purified antibodies from the natural infection could also lead to ASF neutralization levels similar to the recombinant proteins, to the antibodies obtained with the recombinant proteins. So these were promising antigens to use in immunization. Taking this further, the same authors in 1998 cloned P54 and P30 in baculovirus and used this expression in immunization challenge assays. The immunization was made either with P54 or P30 separately, or a combination of both proteins. And a group of animals was also immunized with a low virulence isolate passed in cell culture just to a certain confirmed protection after challenge. When the animals were challenged with Spain 75, the animals that had been immunized separately with the viral proteins showed 0% survival. However, the animals that were immunized with a combination of P54 and P30 did show 50% survival. They two had transient symptoms and one was fully protected. The 3 animals that did not survive also showed delayed disease onset, so partial protection or an effect of the immunization was observed, and all the survivor animals cleared viremia in the end. The proteins P54 and P30 had been observed in a previous study to block virus attachment to host cells, and P30 to block virus internalization. So the combined use of both could have elicited antibodies that by blocking attachment and internalization simultaneously would have led to lower the chances of infection. Of course, this was once again observed in the presence. The immunized animals developed neutralizing antibodies. Later on, a follow-up study by Barderas et al. In 2001 produced a chimeric protein, P54 and P30 fusion, again in baculovirus, and tested again this construct for immunization. And once again, the construct protected the immunized animals from virulent challenge. There was some delayed onset of symptoms and lower viremia than controls, but in the end, the animals cleared viremia. However, in a different challenge model that was done by Nailon et al. In 2004, so the authors used for immunization also a cocktail of baculovirus infected cells producing P30, P54 as the authors before, but also P72 and P22. And in this case, the author challenged the immunized animals with a quite higher dose of the isolate proterozooscope and obtained no survival. There was some delayed onset of disease, but viremia and time course to death was similar to the control animals, in spite, again, of the presence of neutralizing antibodies. So, like Manuel also mentioned before, sometimes when we change the challenge system to another virus, the results are completely different. And there could be strain-specific factors influencing the effectiveness of neutralizing antibodies or other immune responses. At the time that these studies were made, the essentiality of T-cell responses for obtaining protection against ASFV had not been determined yet. So T-cell responses had not been studied at this point. This study came later by Ore et al. In 2005 that showed in a model of immunization with attenuated virus that the presence of CD8 positive T cells was essential for protection. So it would be interesting to know whether T cell responses had been elicited in these studies or not that were made before. Moving on to subunit immunization using DNA. what are nucleic acid vaccines? These are vaccines that introduce in the organism genetic material encoding the antigens. The antigens are produced by host cells and therefore they are correctly folded and have the correct post-translational modifications. In animal studies have shown that they can induce B and T cell responses. And however, the responses sometimes are reported to be too low, but recent advantages have improved the immunization by using adjuncts promoting DNA entry into cells and expression, like using electroporation, codon optimization, or CpG oligonucleotides as adjuvants. The beauty of these vaccines is that they are very easy to produce large scale since they are typically plasmids. The costs are low, they are highly stable, and they can be rapidly also produced. And multiple antigens can be delivered simultaneously as libraries, which is good for screening of protective antigens when we don't know which protective antigens there are. There are no licensed vaccines, DNA vaccines for human use. However, there are several examples that are currently in use in veterinarian field. One of the first studies that was published was by Arguilage et al. In 2011, where the authors cloned P54 and P13 fusion in plasmid, or the authors also tested another construct of P54, P30, and an antibody cloned in sequence with sequence coding for an antibody that could recognize SLA2 expression, therefore targeting the antigens for antigen presenting cells in this way. The authors used the different constructs to immunize animals, and in the first case, in the first simpler construct, there was no response, either antibody or T cell response. And accordingly, upon challenge, there was no protection. With the second construct, when the animals were immunized, there was antibody response. Also against P30 and P54, there was a T cell response that was studied in terms of interferon gamma ELISpot, so interferon gamma producing cells upon stimulation with ASF P30 or P54. And interestingly, in terms of cell responses, also the authors detected an increase in a T cell CD4 positive phenotype in the immunized animals. Non-neutralizing antibodies were observed, and on the contrary, the serum of the immunized animals enhanced the infection in vitro. When these animals were challenged with virulent isolate, there was no protection, and on the contrary, there was even enhancement of disease, and the animals had even higher viremia than control animals. So here the authors thought that maybe this antibody production could be mediating this exacerbation of disease. And this in fact has been observed in other immunizations that I will mention also later on. So this was not the only case where this was observed. And there's also the possibility that the anti-SLA adjuvant or antibody that has been used to target to APC cells could be limiting the T cell response to a T helper phenotype instead of a cytotoxic T phenotype that is believed to be essential for obtaining protection. The same authors tried again a slightly different strategy, this time cloning P54 and P30 together with the soluble hemagglutinin, which is the external part, the extracellular part of CD2V. since this protein had also before shown promising antigenicity properties. One of the constructs was also fused to a sequence coding for ubiquitin. In this way, targeting the antigens directly to the proteasome pathway in cells so that they would be presented in MHC class 1 to cytotoxic T cells. So in this way, skewing the immune response to the desired one. When the first group of animals with the first construct was immunized, the authors saw that they were producing antibodies, and they also had T cell specific responses, only against P30 and P54. No responses were observed against the hemagglutinin, and there were no detectable neutralizing antibodies. Challenged animals with Spain 75 showed no protection and acute disease and viremia, so no protection was obtained. On the other hand, immunization with the second construct, the animals did produce antibody, did not produce antibody responses, which was interesting, and according to the predicted MHC class 1 presentation. The animals did have T cell responses against virus or P30 and P54, although these seem to be slightly lower than the ones obtained with the first construct. And again, no neutralizing antibodies were observed. Upon challenge, however, partial protection was observed. So the animals showed delayed disease, and 3 out of 12 survived. The survivors also cleared viremia 25 days after infection. And interestingly, the authors could see a correlation between survival and the expansion of CD8 positive T cells, so the expected protective phenotype against ASF. So there seemed to be, in this case, partial protection that one speculates could be related to absence of antibodies induced during the immunization. The authors also tested different small Mrna peptides, 9 Mrna peptides, containing in silico predicted CTL epitopes from each of the proteins used in this immunization. And they used them to stimulate PBMCs from the survivor animals. Interestingly, interferon responses were only detected for 2 peptides that corresponded to the hemagglutinin. So the hemagglutinin seems to have CTL or T cell epitopes. A further study later on by Lacaste in 2014 used this time an expression library of random fragments of the ASFV genome covering 70% of the genome, and each fragment fused to ubiquitin. When animals were immunized and challenged, the authors could confirm 60% protection. Although there was transient symptoms and viremia was cleared at 21 days post-infection, which was promising. In order to study the immune responses more in detail, the authors used immunization of serum pathogen-free, specific pathogen-free pigs in order to avoid interference of other potential infections in the immune responses, since this occurs often in outbred farm animals. So the SPF animals were challenged with a cell culture adapted attenuated virus, which was still virulent for the SPF animals. And the authors could confirm similar levels of protection. 50% of the animals were protected. And once again, there were no antibodies pre-challenge. The survivors, once again, showed correlation with this CD8 positive T cell expansion post-challenge. And again, there were also more general T cell responses after challenge, elicited after challenge. So this ubiquitin fusion seems to be determinant and seems to be a very interesting process of inducing protection or partial protection. The author also saw that vaccination with an ASFV library fused to the soluble hemagglutinin was ineffective in protection. So ubiquitin here has clearly a role. However, in such a big library, it's very difficult to ascertain T cell determinants, T cell protective determinants. Regarding viral vectors in vaccination, this has been used for the last 10 years, maybe more. So recombinant viral vectors have the potential for therapeutic use. They enable intracellular antigen expression and induce robust cytotoxic T lymphocyte responses, which can lead to elimination of virus-infected cells. Attenuated vaccine and replication-deficient adenovirus have been used extensively as safe vectors for many pathogen antigens and were observed to induce specific immune responses. There is also robust antibody and cellular immune responses observed in different species. And sometimes an homologous and heterologous prime boost is useful because in this situation, one avoids the recall of the immune response against the priming vector, which can be deleterious for immune responses to the specific antigens. So the first assays that were made to test the immunogenicity of cocktails of adenovirus expressing different viral proteins. So these studies from Locke and Waller et al. In 2016 and 2017 used different cocktails of adenovirus expressing either different structural proteins of ASF or a mixture of structural proteins and other proteins with enzymatic properties that showed promising immunogenicity. The immunization strategy was by prime boost of animals. And when the immune responses were tested, the authors could see that there were ASF antibodies being induced by both vaccinations. And also, I'm not going to go through all the detail, but prime boost responses increased. So the boost was effective in increasing antibody responses. The authors also saw interferon gamma producing cells being induced by both immunizations. These results were then taken further in immunization challenge experiments, where these were published more recently. One of the studies used the previous cocktail of adenoviruses together with two different adjuvants. And the animals were vaccinated and then challenged with Georgia, a virulent isolate. And quite different results were obtained here. So in the group one that have been vaccinated with a mix of structural proteins and other proteins of ASF, they had increased antigen-specific antibodies, but the clinical scores resulted lower than, clinical scores were higher than the control group. So this vaccination was not working. The second cocktail vaccination, which used mostly structural proteins of ASF, the results depended on the adjuvant used, but there was some partial protection observed. The biomice adjuvant induced specific antibodies, high quantity of specific antibodies, but the survival of the animals was low, 2 out of 10 animals. The ZTS adjuvanted cocktail showed more promise with 55% of the animals surviving. However, the results were complicated by the fact that not all the animals in the control groups developed acute disease. So it's difficult to ascertain the correct protection afforded by the immunizations, unfortunately. Similar studies were done later on by Cadenas Fernandez et al. In 2020, where these authors used a cocktail of adenoviruses composed of 35 genes, genes that had known immunogenicity and had predicted T-cell epitopes, and the animals were vaccinated with this cocktail without the biomice adjuvant. This time, wild boars were immunized, and in terms of immune responses, they were poor. There was poorer or absent antibody responses in virus pre-challenge. When the animals were challenged, as would be expected from the immune response, there was no protection, and there was no advantage in using this adjuvant. Later on, not later on, but another study by Jankovic et al. In 2018, published in 2018, used a mixed immunization strategy by using DNA and recombinant vaccinia virus. And the main objective of this study was to determine which epitopes of ASF could constitute T-cell epitopes. So the authors used an expression library expressing 30% of the ASF genome, mimicking the profile of viral proteins during infection. These were in total 47 whole or partial ASF genes. And they used these in prime boost immunization, so using DNA prime and recombinant vaccinia virus boost. Different pools, different combinations were tested. The immune responses were tested mainly cellular immune responses by interferon gamma ELISpot against the specific proteins. And this allowed to establish a rank of antigens according to the response that was induced. Antigen-specific antibodies were also studied and present, but these were variable, and no neutralizing antibody evidence was detected. This study allowed the identification of 14 viral genes, 8 were novel, eliciting good responses. And these results were later on, I will talk about the use of these results in a later experiment that was more successful. The authors also tested 47 antigen formulations in immunization challenge, either in presence or absence of P30. Since P30 is such a strong immunogenic protein, it could skew the immunization towards a more antibody response, which might not be ideal. So in part of the immunized animals, this was avoided. The animals were again immunized by DNA prime and recombinant vaccinia boost and challenged with Georgia virulent isolate. There was no protection observed, and in fact, once again, there was disease enhancement comparing to controls. However, contrary to previous observations, the virus loads in blood and tissues in the immunized groups were lower than in the control groups, which was interesting. Taking this study further, in 2000, very recently published by Gottlieb et al. In 2020, some of the antigens, a subset of the immunogenic antigens from the previous study, were cloned for expression in adenovirus and vaccinia virus and were used in vaccination challenge experiments. One of the experiments tested 2 different pools of 8 antigens, and the most promising responses, the pool with most promising responses were later on used in a second experiment where the animals were immunized with a higher dose and with a pool of 8 of the antigens and also with a subpool of 5 antigens. So the animals were immunized and after immunization showed to have specific antibodies, which were increased by the boost, and also antigen-specific antibodies to variable levels depending on the antigen. Also observed was the presence of interferon-secreting cells, which was in both groups against ASFV and different antigens. Interestingly, upon challenge with virulent virus, it was observed that the first pool conferred 100% protection, which seems to be the best result so far with a subunit vaccine for ASF. The animals, however, had transient clinical signs, but they all recovered by the end of the experiment. However, they did have viremia until the end of the experiment, although the levels decreased along the time. Interestingly, Pool B, with the smaller subset of antigens, conferred no protection to the animals. And just some main conclusions from all these studies. CD2V, P54, and P30 seem to induce protection when expressed in baculovirus. They seem to contain also CTL epitopes. So they can be interesting antigens to include in future vaccination formulations. DNA vaccination by using ubiquitin fusion was also very interesting since it directed immune responses toward the CD8 positive T cell phenotype that is expected to be protective for ASF. And finally, the pools of 8 antigens that were delivered by recombinant adenovirus and MVI were so far the most protective immunization with a subunit vaccine, but such a vaccine is highly complex, and viremia and transient clinical symptoms were observed in surviving animals. So definitely further optimization is needed, although this vaccination strategy seems to be promising, very promising. And I would like to thank you in Purbright, Chris Netherton from ASF Vaccinology, who helped with this review, and colleagues from the ASF group for helpful discussions. Thank you.
Thank you, Rakul. Nice talk, very interesting. I have a question for you. Have you tested these 8 proteins to see if they work against Georgia or produce protection for Georgia?
Actually, yes. This was one recent animal experiment, and the results were not promising. Once again, we see promising results with one challenge model, but when we change to a different virus, things many times don't follow up. And that was also the case. Although there was some protection, still some protection, but not as the one observed that was published.
Thank you.
Can't seem
So far.
to see. Any more questions? I can't seem to see.
There's none in the chat yet. We'll give it a minute, see if
Yes.
anyone's writing. Here we go. In a commercial perspective, which subunit vaccine approach looks more feasible?
In a commercial approach, I would say at this point it's difficult because, for instance, the DNA vaccination that was seen by fusing ubiquitin with the sequences of viral genes, this looked promising, but didn't offer. This would be a very good vaccine in terms of cost, but it did not confer enough protection. So, but maybe optimization in the future could lead to protection. I think this could be an interesting thing to explore. At this point, the complexity of the vaccination with the 8 antigens with recombinant adenovirus and MVA makes it difficult to commercialize, but this will need, of course, further optimization. So attempting to see if we can get protection with less antigens, and also you maybe use different antigens to see if we can also get protection in other challenge models like Georgia, which is the main problem right now, spreading for ASF.
Have you looked into enhancing antibodies for any of the antigens?
Enhancing antibodies, enhancing disease. We still haven't looked. We don't have a clear result for this so far. But this is a problem. So it happens apparently quite frequently in ASF immunizations, and it's something that needs to be taken into consideration. So basically, there could be, if we could circumvent the use of the triggering an antibody response in the different immunization strategies, but apparently antibodies can also offer can also contribute for protection. So this might not be easy to achieve.
Would you recommend to investigate other vectors than adenovirus or vaccine?
I would say this looks quite promising as is. Of course, one can always try to improve, but maybe other vectors that are known to induce disease in pigs, so that we could maybe target immune responses, not only to ASF, but also to another disease. But this is still a bit speculating.
Has anyone tried mRNA vaccines in pigs?
I'm not sure. mRNA per se, I think not. I don't think so.
Okay, and I think it's time for the next talk, and there's no more questions. Thank you, Rakul. Again,
Thank you.
very nice talk.
Bye. Thank you.
Okay, our next seminar will be from Dr. Jan Forth, who for the past 5 years has been a postdoc at FLI in the German National Reference Laboratory for African Swine Fever Virus. His current research interests include African swine fever virus evolution, soft ticks as the arthropod vector, ASF genetics, and next generation sequencing and endogenous elements. Recently, Jan published a paper, a deep sequencing workflow for the fast and efficient generation of high quality African swine virus whole genome sequences in the journal Viruses, where they developed a workflow to get high quality next generation sequencing on African swine fever. Jan will now give his seminar an update on next generation sequencing.
Hey everybody, I hope you can hear me. Thanks for the invitation, happy to be here. To give you a quick overview over NGS and African swine fever, or mainly maybe whole genome sequencing might be a better term actually. So in the end, let's start with a historical view. So this is actually the number of ASF publications over the year. You see there are different events over time. So you see in the beginning, of course, there was the introduction into Southern Europe. Then down here, there was actually the first publication of African swine fever whole genome sequence. You see the eradication of ASF from Europe. And then you see now the new wave starting in 2007, and you see the number of ASF publications per year go up over time pretty quickly. When we now also enter the number of whole genome sequences over time, and this is the accumulated number, you see actually the first sequence, as I said, published in 1995. But since then, actually the number of whole genome sequences increased slowly, but recently quite quickly, let's say. At the moment, we have around 75 ASF whole genome sequences available from GenBank. Actually, I tried to count them yesterday. So this number is relatively new. So the question is, of course, why is this development? Why are the ASF whole genome sequences coming up in the later years more frequently? And of course, you all know the endemic or the epidemic situation of African swine fever, you know, the spread to Asia. So more people are actually affected by African swine fever. And the second thing is the costs have gone down in the last years very quickly for sequencing. Of course, this is for the human genome, but I think you can extrapolate that for sequencing in total. And you see the costs have decreased rapidly over the last years. So sequencing is, for ASF sequencing is getting more cheaper. And since more people are affected, of course, more people are doing it. So more sequences are coming up very quickly in the last years. So why is this, sequencing African swine fever still a challenge? When we look at the ASF genome, you know, of course, it is quite big. It has up to 193,000 base pairs, and it has a lot of genes, open reading frames on both sides of the genome in both directions. It is closed. It has inverted terminal repeats at the end. And it has some other special features. When we look here, this is actually our Illumina reads, MySeq read 300 base per paired end, mapped to a ASF reference, the Georgia 2007 reference. You see here in blue, this is the coverage over the entire genome. So this would be position 1. This would be position 193, 90,000 something. And you see everything where the line goes down, there seems to be a problem. And with ASF, the problem is that has a lot of some complicated motifs for sequencing actually. And one of the most complicated motifs, even for modern sequencing technologies, is actually homopolymer regions. Here you see the Georgia genome has actually regions where it has 17 C nucleotides in a row. And this is actually, as I said, even for modern sequencing, techniques very, very complicated to have a higher resolution in these areas. And these are the regions where you have problems in assembling a high quality genome, where you are sure that you have a correct or at least a near correct sequence. So let's have a look. How is the situation on ASF whole genome sequencing? Sequences available in 2011, actually, we had from 24 genotypes that we know of today, we had actually 9 available, and you see most of them actually were published around 2003. This was at this time the newest one, the Georgia sequence, which was done in Pirbright. And since then, actually something happened. Let's go through the years. In 2014, you see actually some new sequences came online, but you still see there's actually only one new genotype. Most of the new sequences that came up are on the known genotypes 2 and 1, which were, of course, the ones that were pandemic. In 2015, you see actually it's only a few additional sequences coming up. 2016, only one. 2018, coming up a little more. And now you see actually, one of the problems I will talk in a few seconds, but the genotype 2, of course, which is of broad interest because this is the one actually spreading in Eastern Europe and Asia. More and more sequences are coming up there. This is the situation 2019, actually some new strains, but again, mostly genotype 2 sequences coming up. This is the situation at the moment. You see again, a lot of genotype 1 and genotype 2 sequences, very few of the other exotic genotypes. So at the moment we have around 10, a little more genotypes from 24, but we are a bit far away from actually knowing most of the genotypes that are around. So what is the problem? The main problem is actually at the moment, we have a lot of genotype 2 sequences from Eastern Europe and Asia. They're only, over the last 10 years, they're actually more than 99% identical to each other. So they only differ in single nucleotides over the entire genome length. So we were not able to actually do some kind of molecular epidemiology. We do no idea on conclusions on origins of introductions. For most of this, very, very few variations we see, and we have no correlations on the gene or the protein function, or if there is some influence, because we don't know the function of most of the genes, as actually was told earlier this day. One good example is the ASF Belgium sequence. We actually did this, and we had 15 single nucleotide differences to the Georgia reference, which is actually 10 years older. Quality control on the sequences. So we have with most of the, some of the sequences, I have to say, we have no metadata to validate if the sequence was done correctly or to reanalyze it to see if we see differences. And I mean, a single nucleotide difference is very, very easily mistaken, for example, by assembly mistakes or by mapping artifacts. So this is actually a big problem at the moment with the genotype 2 sequences. So we desperately need new strategies.
Jan, we can't hear you.
Can you hear me now?
Yep, that's better.
Okay, sorry. So you see on the left the phylogenetic tree, and you see on the right the map, and you see every color here is a dot on the map. And what you can see is that, okay, the genotypes are pretty well distributed over Africa, so we have some information. But when we now subtract all the sequences we have only from the domestic pig population, you see actually we know pretty, we have pretty few information on the actually sylvatic cycle. We have a few, but in the end there's a sylvatic cycle all over Southern Africa. So there's actually information missing. So cooperation with researchers from Africa is actually very crucial here in this point to get together the resources, the strains that are available, and on the other hand, the technique, the methods, the people who can sequence, and also have the bioinformatics background and also the background in ASF sequencing and bioinformatics to get this sequences done. So what is actually now in progress at the moment? On the left side, you see a P72 phylogenetic tree. So these are actually all the genotypes that are around at the moment. And this is actually on the right what at least we have in progress at the moment. And you see we have in cooperation with Chris from Pirbright and also with Edgar Zimbulundu and Herman Chambara from Zambia, we have produced more whole genome sequences from different genotypes. We are still in the progress of validating some of them, adding more different sequencing approaches to validate the sequences, but we are pretty far along. So I hope we will be able to publish some of these sequences soon and actually expand the knowledge on different genotypes, on different setups of the ASF genome. And I can tell you there are main differences between the different genotypes. So what's also, of course, necessary and also very interesting is we need to identify interesting targets. So we need to go away from sequencing random strains of genotype 2, like say, okay, I have to sequence 20 isolates from Estonia or from Latvia or wherever. But we need to detect more specific, to pick more specific our targets for sequencing. Very good examples is here. We need to detect and analyze the rare variants that actually exist. For example, the Estonia variant, which was sequenced in 2014. It actually is a field virus, so it was isolated in the field, and it has a 14 kb deletion at the 5' end and some reorganization of 7 kb, and it has an attenuated phenotype. So this is actually something we can learn something from. We want to actually identify these. So what we need actually would be some kind of whole genome-based screening for ASF genotype 2. So have a method, a very easy method to just have to see if everything is there, is there some reorganization or something. And if we see something interesting, then we go a little deeper and say, okay, now we do deep sequencing. Now we look a little more specific into what's going on there, but without investing so much money and time in the first place to sequence strains that are almost identical. What we also need very urgently, and this is also something that Manuel said earlier, is of course, we know there is changes in cell culture adapted viruses, and we also have these, we've seen this in the past, sorry, on WSL cells, for example, there is adaptation. These viruses tend to lose genetic information, actually pretty extensive genetic information, around 20-25 kbs. So this is actually something we need to be aware of, and also we need to check regularly these isolates when we work on them on cell culture to see if something else happened, if we get off-target effects, or if we get something we didn't expect. Also very important, I think, is the part on resequencing old virus isolates. So sequences that are in the database for a long time. We actually had this with the Georgia 2007 sequence, which we resequenced and corrected a bunch of actually sequencing errors, which were introduced by an older sequencing method. And also database curation. This is a very important point because the sequences are used by many people to produce to set up their experiments to build on their research. So they actually should be done with the most sophisticated methods we have to see that they are correct and that people are working with correct information. And of course, what we need, and this is actually very urgent, we need transcriptome sequencing. So we need to have updated annotations. We need to know what is going on at transcriptome level. What are the difference between different viruses? because there's actually a gap in knowledge. And we have now some transcriptome analysis coming up from Pirbright. We have done some which might be published soon. So actually there is some information now, but we need we need more of that. Also very interesting topic is quasi-species or variant analysis. There is not much known on ASF on that part. But from time to time you get some not evidence, but some information on there. There might be actually something going on, but we are not very far along there. So do we need more methods? Well, we have for example, this is only an example, the Illumina technology. We use mostly the MiSeq because we have it available, but most of the Illumina machines are very well suited for ASF sequencing. They have a high accuracy, they have a high output. You can do multiplex sequencing. Sorry. But of course, the read length is pretty short. You have problems with homopolymer errors, especially also with the Illumina platform, but especially iTorrent is very error prone in homopolymer regions. And of course, you have a long run and prep time, especially for the Illumina systems. In combination, of course, with other methods, for example, the Oxford Nanopore MinION, which is a third generation sequencing method. It produces ultra long single molecule reads. It has a very short runtime, a very short sample prep time, but of course some disadvantages. The accuracy is low, the output is low, and you need a high quality DNA sample actually. But in the end, put them together, and this is actually the point. So we need to combine the methods we have. We have enough good methods for sequencing, but we need to combine them. We need to be smart about that to get what we want in the end. And at the moment, we want high quality ASF whole genome sequences. So whatever, in the end, a huge problem, almost always the huge amount of host sequences in the background. So usually when we sequence specific let's say like tissues from infected swine, we are talking about 0.01 to 0.05% of viral reads per sample. This is actually, you have to do very large data sets to get enough reads to build your whole genome sequence. So, but also we have methods for that. For example, at the FLI, we use target enrichment. So you just capture out what you want to sequence, you clean it up, you amplify it, and then you sequence especially what you want to sequence, in this case, the ASF reads in your sample. Other people doing it the other way around, to get rid of all the host sequences and sequence whatever is still is in the sample. This is also a good way. Both actually in the end bring you a removal of background sequences. You can do high coverage sequencing. You do not need to do as much reads, so you do not need to have much resources, computing power, and also money, of course. But of course, there's also some disadvantages. Here's amplification bias. It is pretty expensive, especially when you do the MyBaits capture or the target enrichment we do. And of course, with our methods, for example, you only capture known targets. So in the end, we have good sequences, we have good methods. The thing is, we want the quantity of the sequences. We want more sequences in the database, and there's where the quality part comes in. We need metadata and we need quality control on the sequences that people produce. So we need raw reads, we need coverage information, we need information about what tools we used, what platforms were used, and so on and so on. So the researchers actually can validate the sequence they find in the database and say, okay, I trust that sequence because I can see, okay, it was done from this and this. I can actually repeat the analysis if I want to. We have good methods, as I said, but we need to be smart about combining these methods actually to get, in the end, what we want, a good high quality sequences. And of course, what would be extremely helpful, especially in Eastern Europe and Asia, is the whole genome-based screening approach. So maybe, for example, combining Illumina with target enrichment with the MinION platform, for example, to fish out of a sample what you want to sequence, put it on a MinION in a very quick time, have a draft genome sequence where you see, okay, what is in and what is not. And of course, and this is also most important, I can suggest that enough is we need to have improved sample selection. So we need more genotypes. So we need to have cooperations with people in Africa, in Eastern Europe, wherever ASF is going around and something interesting's come up. We need to have more information on the Slovatic cycle, but probably is where all these genotypes are actually coming from. And of course, and this is, well, one point I have also to stress is we will, of course, do a whole genome sequence when we have a new introduction. So we, of course, want to know.
We lost you again, Jim.
Sorry, it's this button here. So in the end, we will still sequence new introductions into new countries, but yeah, we have to do that with a little more specific background. So with that, thank you for your attention, and thank you for the opportunity to say something about sequencing here. I'm happy to answer some questions. Thank you.
Thank you, Jim. Nice talk. I have a question for you. What do you think the value of genotyping as currently established in African swine fever?
You're referring to the P72 genotyping?
Yes.
Well, I have mixed feelings about that. On the one hand, of course, it's useful up to a specific end because you can have, with a very simple tool, have some kind of idea where a virus might belong to. But in the end, when you compare this with the whole genome information, you sometimes get a different picture. And this is actually what we have now, what we will see in the future with more genotypes coming up, that there are discrepancies between the whole genome information and the P72 information. So I think we will have to think about maybe new method or improved or modified method for genotyping in the future. But in a moment, I think it is how it is.
Next question is there evidence for recombination between genotypes in areas where several genotypes are present, such as Africa?
Actually, it is probable. There is some evidence that there might be actually, most probably in the Slovatic cycle, there might be some recombination. It might be actually the tick that is involved here. I know from no reports from domestic pigs or from other swine that there is recombination, but in the tick vector, it might be actually possible. Yeah.
Okay. Next question is, Estonia 2014 seems to be significantly different from all other genotype 2 genomes. It was sequenced at FLI and published in 2018. Can you please elaborate on the rearrangements you observed there? Have you tried to resequence it to confirm these rearrangements and deletions?
I'm happy to. And yeah, we actually sequenced that in 2018, I think. It came from Estonia. It was a field virus which went here into animal trials. What we found is actually, it was also for us pretty surprising because we found a 14 kb deletion on the 5' end. But also we found a 7 kb duplication, so 7 kb from the 3' end were actually invertedly bound at the 5' end. So there was a deletion and there was a recombination at the same time. We sequenced that several times. We resequenced that. We did PCR on that. We sequenced the PCR amplicants because we also wanted to be sure that this was actually accurate. But in the end, yeah, we came up with every time the same thing. The recombination was there. And yeah, it's a very interesting virus.
Next question is, Estonia 2014 is attenuated. Have you done animal experiments?
Yeah, we have. Actually, it was published. Several experiments were published, also in the paper where the sequence was published, actually. So yeah, we have, and it seems to be attenuated to some point. At least more animals survived that with the wild type. Yeah, but in the end, and this is, of course, this is important, Sandra just intervened. It has not actually been, it is not in the field anymore. So actually it was probably overgrown by the wild type. We don't know why for sure, but there are some speculations around. But in the end, it is a attenuated virus, so it probably does not kill as effective as the wild type. And since ASF needs to spread through blood and tissues of, for example, dead animals, also bleeding animals, probably it was just overrun by the wild type. But this is actually just a speculation. Yeah, but in the domestic pig, it would be not nice, yeah, because of the attenuated phenotype, of course.
All right, that looks like all the questions. Oh, no, one more. Besides Estonia and LV17, have you found more natural attenuated strains in Europe?
So far, I'm not aware of more actually. We actually, at least, have not found more attenuated strains. I think these are the only ones. Yeah, exactly. 2, I think, yeah. No, I'm not aware of more.
You mentioned a simple technique to screen for genotype 2 viruses and make sure they differ from the ones we know before going for whole genome sequencing. Can you please elaborate on that screening technique?
Actually, at the moment, it's more a concept. It's not a specific technique, but at least it's the combination of 2 techniques. So we have, for example, the capture, so the target capture of specific sequences from a sample, and we have the Mrna sequencing. One, actually, the target capture produces very pure samples of what you want to sequence, so the ASF sequences. And the other one, the Mrna platform, produces a very simple and quick tool to get sequencing done. Of course, the Mrna has a problem on its own. It's not very accurate. But in the end, if you have a high coverage, you can actually make up for the low efficiency of the sequencing. So in the end, the combination of both could be a valuable tool to get a whole genome screening approach. Another approach would be to combine, for example, the capture with Illumina iSeq technology, which is a very small 1 cartridge system where you could insert a lot of captured samples at once, reducing the price significantly for building one specific ASF genome. And using this just for a screening approach, and then in the end, going for deep sequencing on interesting samples. But this is, at the moment, it's more a concept.
Any advice to use Mrna for ASF sequencing?
Well, depends on the question. If you want to do it for whole genome generation, usually you need high quality DNA. So prepare good DNA from your sample. If you want to deploy it in the field, I think this was actually a question that was raised earlier. Of course, it could be used in the field. The question is, it would have to be validated very good to see if the information you get from a field sample in the field actually can live up to the standards of a high quality whole genome sequence. But this in the end depends on the question you have. Do you want to have this from the field, or is it just you want to have some basic information in the field very quickly and then go to the lab and do the more sophisticated analysis there? That depends entirely on the question.
Do you know the background of the strains that was ever used as a vaccine in Spain and Portugal around last century, 1960s?
Not specifically, no. I have not looked in on that into detail. No.
Have you tested to remove the host genome by using enrichment towards microbiome kits?
We have not so far tested that because the capture is working very well. I know another technique that was used to remove the host sequences. I think methylation was the key point here, but we have also not tried that because usually for us the capture is working pretty well.
Okay, I think that is all of the questions. With that, I think it's time to thank all the speakers. We had a lot of good talks today. And again, to thank the University of Pretoria for hosting the webinar. And we will see you guys hopefully all in Uganda in 2021.